Hepatic encephalopathy is a condition where liver dysfunction leads to buildup of toxic substances like ammonia that affect the brain, causing confusion and altered mental status. Now, many of these toxins are actually produced by bacteria in the gut. So when doctors gave oral antibiotics, they reduced the number of these harmful bacteria, which lowered toxin production. As a result, patients' brain symptoms improved. Since then, research has shown that any imbalance in gut microbiota can disrupt gut-brain communication. Think of the gut-brain axis as a constant two-way conversation between the brain, mainly the central nervous system, and the gut's own nervous system called the enteric nervous system. On one hand, the brain can influence gut functions like motility, secretion, and even the environment in which gut bacteria live. On the other hand, signals from the gut, especially from the gut microbiota, can travel back to the brain and affect emotions, behavior, and thinking. This communication between the gut microbiota and the brain does not happen through a single pathway. Instead, it occurs through multiple interconnected systems, including nerve pathways like the vagus nerve, endocrine signals, that is through hormones, immune responses, and various chemical messengers circulating in the blood. So in simple words, the gut-brain axis is a two-way communication system in which the brain and the gut microbiota are constantly interacting with each other. Any changes in the gut microbiota can influence brain function. At the same time, the brain can also shape the gut environment with the help of these above pathways. Now, importantly, our body is not just human cells. It is actually a complex ecosystem filled with trillions of microorganisms living on the skin, in the mouth, and most importantly, in the intestines. These microorganisms, often called commensals, are not harmful. Rather, they play a key role in maintaining normal body balance or homeostasis. The proof that the gut-brain axis exists comes from multiple types of studies. For example, in animal experiments, when scientists change the gut bacteria, either by giving antibiotics or transplanting microbiota, the animal's behavior also changes, such as showing more anxiety-like or less social behavior. In humans, people with conditions like irritable bowel syndrome often have higher rates of anxiety and depression, showing that gut problems and brain symptoms commonly occur together. There are also studies where giving certain probiotics leads to small but measurable improvements in mood and stress levels. On the other side, stress and emotions can directly affect gut function, changing motility, secretion, and even the composition of gut bacteria, which is why people feel butterflies or develop diarrhea during stress. So with these evidence, we have strong proof that the gut and brain are constantly influencing each other in both directions. To understand the functional neuroanatomy of the gut-brain axis, think of the gut as having its own mini-brain called the enteric nervous system, which can function independently even without input from the central nervous system. However, under normal conditions, it remains in constant two-way communication with the brain. This communication is regulated by the autonomic nervous system, which has two main components, the parasympathetic and sympathetic systems. On one hand, the parasympathetic system, mainly through the vagus nerve, stimulates gut activity. When cholinergic signals travel from the brain to the gut, they act on two important nerve networks. First, the myenteric plexus to increase gut motility, and second, the submucosal plexus to enhance secretions. On the other hand, the sympathetic system through the splanchnic nerves does the opposite by reducing gut motility and decreasing blood flow to the intestines. Meanwhile, within the gut itself, the enteric nervous system coordinates local functions like secretion, movement, and blood flow through its internal network of neurons, while also sending sensory signals back to the brain. So, in simple terms, the gut can function on its own, but it is constantly being fine-tuned by signals coming from and going back to the brain, creating a tightly regulated and dynamic communication system. Now think of the gut microbiota as a huge community of microorganisms living inside our body, especially in the intestines. These include bacteria, viruses, fungi, and even some parasites, and they can be helpful, harmless, or sometimes harmful. Now, importantly, we don't just carry these microbes. We actually live in a symbiotic relationship with them, meaning both we and they depend on each other for survival. In fact, the number of microbial cells in our gut is even greater than the number of human cells in our body, and together they weigh around 1 to 2 kilograms, which is roughly equal to the weight of the brain. Escherichia coli was first identified in healthy individuals, and later the term microbiota was introduced to describe this entire ecosystem. Now, this microbial population doesn't appear randomly. It actually starts developing from birth, and its composition is influenced by several important factors. For example, whether a baby is born through normal vaginal delivery or cesarean section, whether they are breastfed, exposure to infections, use of antibiotics, diet, and even stress. All of these shape which microbes grow and thrive in the gut. Now, when we talk about how the gut and brain communicate, this interaction occurs through four major pathways: the neural pathway, enteroendocrine signaling, the serotonin and tryptophan pathway, and the immune signaling pathway. Let's first start with neural pathway. Think of the neural pathway as the fastest and most direct line of communication between the gut and the brain, mainly through the vagus nerve. Now, during stressful situations, when the body senses a threat to its balance, that is, homeostasis, the brain can quickly adjust gut function by sending signals to the enteric nervous system. At the same time, this communication is not one way. Signals also travel from the gut back to the brain. Interestingly, certain gut bacteria can influence this signaling. For example, Lactobacillus rhamnosus can send signals from the gut to the brain through the vagus nerve. These signals can affect certain receptors in the brain called GABA receptors. Now, what are GABA receptors? GABA is the brain's main calming neurotransmitter. Its job is to slow down brain activity and prevent overexcitation. So when GABA binds to its receptors, it produces a relaxing anti-anxiety effect, basically telling the brain to calm down. So, when lactobacillus influences these GABA receptors, it can enhance this calming effect, which is why it is linked to reduced anxiety and depressive behavior. Similarly, in conditions like colitis, another bacterium called Bifidobacterium longum has been shown to produce an anti-anxiety effect. But importantly, this effect only occurs if the vagus nerve is intact. So in simple terms, the vagus nerve acts like a communication highway, allowing gut bacteria to directly influence brain function, especially emotions and stress responses, showing how closely connected our gut health is to our mental well-being. Second is enteroendocrine pathway. Think of enteroendocrine signaling as a chemical way in which the gut talks to the brain. Now, when gut bacteria break down food, they produce certain byproducts. And when these come in contact with the lining of the gut, they stimulate special cells called enteroendocrine cells. In response, these cells release small chemical messengers known as neuropeptides. These neuropeptides then act in two ways. First, they work locally within the gut by influencing the enteric nervous system, helping to control things like gut movement and secretion. At the same time, some of these peptides enter the bloodstream and travel to the brain, where they can influence functions like appetite, digestion, and even mood. Examples of these important neuropeptides include peptide YY, neuropeptide Y, GLP-1 and 2, and substance P. So these gut bacteria actually play a major role in controlling how much we eat, how well we digest food, and even how we feel by these chemical signals. Now third is serotonin and tryptophan pathway. Serotonin is a key neurotransmitter that affects both the gut and the brain. Interestingly, about 95% of serotonin in the body is actually made in the gut, mainly by special cells called enterochromaffin cells and by neurons of the enteric nervous system. Now, in the gut, serotonin helps control important functions like movement of the intestines, secretion of digestive fluids, and even pain sensation. On the other hand, in the brain, serotonin plays a major role in regulating mood, emotions, and thinking. Furthermore, certain gut bacteria, especially spore-forming bacteria, can promote the production of serotonin from tryptophan. Tryptophan as an essential amino acid, which means your body cannot make it on its own, so you must get it from your diet. Common sources include foods like eggs, milk, cheese, chicken, fish, nuts, and seeds. Now, once we consume tryptophan from food, it reaches the gut. Here, gut bacteria, especially spore-forming bacteria, convert tryptophan into serotonin, particularly in the gut's enterochromaffin cells, thereby influencing how much serotonin is available. Because of this, any imbalance in these bacteria can affect both gut health and mental health. For example, altered serotonin signaling has been linked to conditions like inflammatory bowel disease as well as mood disorders like depression. So in simple terms, the gut produces most of the body's serotonin, and with the help of gut bacteria, it can influence both digestion and how we feel mentally. The last one is immune signaling pathway. First of all, the gut contains a very large part of the body's immune system called gut-associated lymphoid tissue, which acts like a security guard lining the intestines. Its job is to constantly check everything that enters the gut, like food, bacteria, and other substances, and decide what is safe and what is harmful. Now, when harmful bacteria or infections are detected, this system gets activated and starts an immune response, releasing signals such as cytokines to fight the threat. However, these cytokines don't stay only in the gut. Some of these cytokines, such as interleukin 1, 6, and TNF-alpha, can cross the blood-brain barrier, while others can activate sensory afferent fibers of the vagus nerve, which serve as a direct communication pathway from the gut to the brain. As a result, the brain responds by altering behavior, which is why infections can make a person feel anxious, low, tired, or generally unwell. On the other hand, not all microbes are harmful. In fact, beneficial gut bacteria produce substances that help regulate the immune system. For instance, they can increase T regulatory cells, which are important for controlling excessive immune reactions. And they also promote the release of anti-inflammatory cytokines like interleukin 10, which help calm inflammation. Studies have even shown that consuming beneficial bacteria like Bifidobacterium infantis can increase interleukin 10 levels in humans. So the gut microbiota helps balance the immune system. So these are the four main pathways through which the gut microbiota communicates and connects with the brain. Now, after understanding the four main pathways, let's see how the gut-brain axis actually helps maintain normal health. First of all, the presence of gut microbiota from early life is very important for the proper development and maturation of both the enteric nervous system and the central nervous system. In fact, if these microbes are absent, it can lead to imbalance of neurotransmitters in both the gut and the brain. As a result, normal gut functions like movement and coordination get disturbed, leading to problems such as delayed gastric emptying and slow intestinal transit. And in the brain, this can affect mood, behavior, and cognition. For example, increasing the risk of anxiety, depression, poor stress tolerance, and even problems with memory and learning. Furthermore, the gut microbiota also plays a key role in controlling how the body responds to stress. It does this by influencing the hypothalamic pituitary adrenal axis, which is the main stress response system. When the gut bacteria are balanced, they help keep the HPA axis under control, so the stress response is appropriate and not excessive. But when there is dysbiosis, that is imbalance of gut bacteria, for example in condition like IBS, this system can become overactive, leading to higher levels of stress hormones, cortisol. This is why changes in gut microbiota have been linked with increased stress, anxiety-like behavior, and even mood disturbances. In addition, these microbes help maintain the mucus layer and biofilm in the gut by secreting substances like acids, bicarbonate, and mucus. This is important because it supports proper fluid balance in the intestines and strengthens immune defense at the gut lining. Now, after understanding how the gut-brain axis supports normal health, it becomes easier to see how its disturbance can lead to disease. In fact, the role of gut microbiota in disease was first noticed when antibiotics were used in conditions like hepatic encephalopathy. Hepatic encephalopathy is a condition where liver dysfunction leads to buildup of toxic substances like ammonia that affect the brain, causing confusion and altered mental status. Now, many of these toxins are actually produced by bacteria in the gut. So when doctors gave oral antibiotics, they reduced the number of these harmful bacteria, which lowered toxin production. As a result, patients' brain symptoms improved. Since then, research has shown that any imbalance in gut microbiota can disrupt gut-brain communication. As a result, this disturbance has been linked to several conditions like depression. Studies have shown that people with depression often have an altered composition of gut bacteria, such as changes in Acinetobacter and Bacteroides. Interestingly, when microbiota from depressed patients are transferred to healthy mice, those mice begin to show depressive-like behavior along with changes in brain activity, especially in areas like the hippocampus. Furthermore, depression is also associated with changes in the immune system and brain structure. The immune system can become altered, and important brain chemicals like serotonin and brain-derived neurotrophic factor are often reduced. At the same time, certain parts of the brain, such as the amygdala, can undergo structural changes. So depression is not just a mood problem. It involves changes in brain chemicals, immune function, and even brain structure. In addition, gut microbes produce important substances called short-chain fatty acids, such as acetate, propionate, and butyrate, which can influence body functions. For instance, increased acetate production can activate the parasympathetic nervous system, leading to increased secretion of insulin and ghrelin, a hunger hormone. As a result, this may cause increased appetite, weight gain, and metabolic changes, which are often seen alongside depression. So altered gut microbiota can influence the brain and contribute to depression by affecting brain chemicals, immune responses, and metabolism. Now that we understand depression, let's look at another condition linked to the gut-brain axis: irritable bowel syndrome. In IBS, changes in the gut microbiota can disturb the normal regulation of the hypothalamic pituitary adrenal axis, leading to increased release of stress hormones like corticotropin-releasing hormone. It then acts on the pituitary gland to stimulate the release of ACTH, which in turn acts on the adrenal glands to increase the production of cortisol. This increasing corticotropin-releasing hormone and cortisol, which makes the gut more sensitive and often more active, contributing to diarrhea. At the same time, there is also dysfunction of the autonomic nervous system. In IBS, there is often reduced parasympathetic activity, meaning the rest and digest signals are weaker, along with irregular sympathetic activity, which disrupts the normal rhythm of the gut. As a result, during periods of stress or increased corticotropin-releasing hormone activity, the gut becomes overactive, leading to increased motility and diarrhea. On the other hand, when parasympathetic activity is low and sympathetic tone becomes relatively dominant, the gut becomes sluggish, leading to constipation. Because this control is unstable, the gut keeps shifting between these states, which explains the alternating pattern of diarrhea and constipation seen in IBS. Furthermore, IBS is associated with low-grade inflammation in the gut, meaning the immune system is mildly activated, which further alters gut movement and increases pain sensitivity. Interestingly, brain imaging studies have shown that certain areas of the brain, such as the anterior cingulate cortex, prefrontal cortex, and insula, are more active in IBS patients. And these regions are involved in processing pain, emotions, and gut sensations. So in simple terms, IBS is not just a gut problem. It is a gut-brain disorder, where stress, altered microbiota, immune activation, and brain changes all work together to produce the symptoms. Now, after IBS, let's look at autism spectrum disorder in relation to the gut-brain axis. Autism is a neurodevelopmental condition where children have difficulty with social interaction, communication, and often show repetitive behaviors with varying levels of intellectual ability. Interestingly, many children with autism also have gastrointestinal symptoms like constipation, diarrhea, and feeding difficulties, and these often become more severe as the autism severity increases. Now, apart from genetic and environmental causes, research suggests that altered gut microbiota and oxidative stress may also play an important role. Studies have shown changes in gut bacteria, such as increased ruminococcus and Bacteroides, and decreased firmicutes. At the same time, stool samples of children with autism often show reduced levels of short-chain fatty acids like acetate, propionate, and butyrate, which are important for gut and brain health. In addition, certain harmful bacteria like Clostridium tetani and Desulfovibrio may worsen symptoms. Furthermore, interventions like probiotics and a gluten-free diet can help modify the gut microbiota, improve gut immunity, and may lead to better symptom control in some children. Finally, it's important to note that the gut-brain axis is not only linked to autism, but is also being studied in other neurological conditions like multiple sclerosis, stroke, Parkinson's disease, and Alzheimer's disease, although research in these areas is still ongoing. So, in simple terms, autism is not only a brain disorder, but also has a strong gut connection, where changes in gut microbiota may influence both gastrointestinal and behavioral symptoms. In conclusion, current evidence clearly shows that the gut microbiota plays a central role in the two-way communication between the gut and the brain. So factors like stress and excessive or improper use of antibiotics can disturb this microbial balance and contribute to disease. Therefore, maintaining a healthy lifestyle and balanced diet, along with the judicious use of antibiotics, is essential for keeping this gut-brain connection healthy. Furthermore, controlled ways of modifying the microbiome are emerging as a promising area of research, which may offer new treatment options for conditions like IBS, multiple sclerosis, Parkinson's disease, and dementia.